Good morning, dear students. Welcome back to our official YouTube channel, SRN BGNR Government Arts and Science Degree College, Khammam. And this is Dr. B. Satish Kumar, Assistant Professor in Biochemistry. And now we are learning about the molecular biology processes. And in last two classes, video lessons, you have learned the basics of the regulation of gene expression, in which we covered the following syllabus. So the third unit of molecular biology paper is regulation of gene expression. Okay. So the syllabus covered so far is the induction and repression. The first subtopic of this unit is induction and repression. So we have already discussed about what is induction and what is repression. Okay. So induction means here the genes, in fact it is about the regulation of gene expression. Gene expression is nothing but transcription plus translation. Okay. So from the gene, a protein product, the purpose of the gene, the very purpose of the gene is production of a polypeptide chain, functional polypeptide chain that participates in different functions in a particular function. Like if it is an enzyme, then it participates in a chemical reaction of any particular metabolic pathway, or if it is a receptor protein, then it binds a particular hormone or that ligand molecule and carry out, convey the signaling process. And otherwise, if it is a structural protein, so that gives strength to the supporting structures like muscle, skin, and bones, like etc. So we have several different functions for the proteins, because proteins are really very they have diversified functions. In case of carbohydrates, lipids, and nucleic acids, we have only limited number of functions, but in case of proteins, the list is very vast, and very diversified functions are carried out by the proteins, because uh, um, it uses 20 different amino acids, and all the 20 different amino acids are different from each other in their physical and chemical properties. Okay. So mainly the importance of proteins, the diversity in the function of the protein is because of their dynamic nature. So the proteins can act as structural component, in which the proteins do not change their conformation or shape, just like rigid structures they form. And as well as structural role, it has some dynamic role, like enzymes, antibodies, and receptor molecules, and so actin and myosin, like uh, proteins which participate in muscle contraction. So many different functions are there. So the conformation changes according to their function. Okay. So that is why proteins have really very diversified functions, and because of the existence of the proteins only, the diversity of life has taken shape. So that is why the proteins are prime molecules when we compare all the biomolecules. Okay. So the ultimate purpose of gene itself is production of a polypeptide chain. So that therefore the information within the gene is translated into the amino acid sequence of a polypeptide chain. So in that we have two stages, transcription and translation. So we have already learned, you have already learned about the whole process of transcription and then translation. So what happens during the transcription, how mRNA molecules or RNA molecules formed from their corresponding genes, and in the translation you have learned how the mRNA participate in the translation process, and how a polypeptide chain is synthesized based on the information present in the mRNA molecule. Okay, so that is the expression, gene expression, and regulation of gene expression is essential thing, because all the genes must be regulated. Okay, otherwise if there is no regulation, then the functions may not be coordinated, and the organism itself, it is an organization. So each and every function must be coordinated with each other, and any particular time only needed genes should be expressed, and remaining genes must be repressed, because they are not needed at that time. They may be needed after some time, or when based on the conditions and surrounding environmental factors. Some enzymes, some genes must be positively regulated or expressed, and some must be repressed. Okay, so in that context we have learned about two processes like induction and repression. So in case of induction, we have studied the example of lactose operon, and in the repression we have studied about the repression process by taking example tryptophan operon as an example. Okay, so now let us quickly just recap the induction process and the repression process. Okay, so in induction process, the operon the purpose of the lactose operon is to take up the lactose in the medium and then break the lactose. Lactose is a disaccharide containing glucose and galactose. Okay, so to utilize that lactose as energy source, the bacteria must hydrolyze the glycosidic bond between glucose and galactose. Okay, first of all, it should allow the entry of lactose into the cell from the surrounding medium. Then once it is inside, it should be hydrolyzed to give the constituent sugars like glucose and galactose. So therefore it must carry out two functions for the utilization of lactose by the E. coli cell. Okay, so here these functions are carried out by their particular proteins and enzymes. Okay, the hydrolysis of lactose is carried out by an enzyme called beta galactosidase, and the uptake of lactose inside the cell requires a permease protein, that is not an enzyme, that is a transporter protein, and that allows the entry of lactose into the cell. Okay, so in both the cases, both these proteins are they must be synthesized when the lactose is present in the medium. Then only they must be synthesized. Otherwise the synthesis of beta galactosidase and permease proteins in the absence of lactose also, then what happens? Most of the energy will be wasted for the production of these unuseful proteins because they are not necessary at that moment when the lactose is absent. So if the lactose is not present in the medium, then operon must be switched off. Otherwise the cell will cell is going to synthesize those proteins which are absolutely not necessary. Okay, so to conserve the energy, it should be regulated. So for that purpose, the regulation of lactose operon depends on presence and absence of the lactose. So lactose itself switches on the lactose operon. So that means the lactose operon will always be in switched off form. Once we add lactose to the medium, then only that lactose operon will be switched on by the lactose itself. So that is why lactose acts as an inducer, and that induces or switches on, it switch on the lactose operon. Okay, therefore it comes under the induction process. Okay, so we have already seen that lactose operon contains 3 structural genes, ZYA, Z that encodes a polypeptide chain or a protein, that is nothing but the beta galactosidase enzyme, and LAC-Y gene is responsible for the production of galactosidase permease that allows the entry of lactose into the medium, and LAC-A gene, that is transacetylase. So this is an enzyme that detoxify the unwanted materials, unwanted molecules which entered into the cell along with the lactose through the permease. Okay, so any unwanted toxic materials if enters into the cell along with the lactose, so they must be cleared off from the cell, otherwise they cause harm, they may harm the bacteria. So that is why the unwanted molecules which enters bacteria should be detoxified. So for that purpose it has this particular gene also. So this gene will detoxify. Okay, so the main, these are 3 are the structural genes, and one main gene that involve in the regulation of lactose operon is LAC-I gene. I stands for induction here, induction. So that is why I gene, in fact it is a regulatory gene. Okay, this is a gene in regulatory sequence. In control sequence we have a gene and 2 regions, promoter site and operator site, because in fact the promoter site is a specific sequence, that is not a gene, a specific sequence, and operator is also a specific sequence. Okay, protein binding sequence. But in case of regulatory gene, that regulatory gene is a gene which has its own promoter, pi itself is a own promoter. This regulate the structural genes, but there is no regulation for this gene, in fact. Okay, so you have to remember this. Okay, this gene will be constitutively expressed. Okay, whether the lactose is there or not. If the lactose is present in the medium or absent in the medium, the gene the LAC-I gene always in the switched on form, but it produces at a very slower rate, expresses at very slower rate. So less quantity of repressor will be produced continuously. Okay, so if the lactose is absent, then what happens? The LAC-I gene is continuously producing the repressor molecule. So that repressor protein which is active active protein. So that active protein, the function of the active protein is it binds to the operator site. Okay, it recognizes specific sequence at the operator region and it binds there. Okay, it, then because of that binding, it occupies the entire operator sequence. So now the RNA polymerase, which has to bind to promoter region for the transcription process, but in this case the repressor is already occupied by the operator, and that prevent the binding of RNA polymerase to the promoter region. So because of the occupied position of operator sequence, the promoter sequence cannot bind RNA polymerase. Even if it binds to the promoter, it binds very loosely, and at the same time it cannot move forward because the operator is bound with repressor molecule that is present in front of the promoter sequence. So this is the position of operator is also very important, because it is just downstream to the promoter. Operator's region is located that downstream to the promoter. So that is why that will obstruct the movement of RNA polymerase and stop it. Okay, if there is no repressor molecule on the promoter, then what happens? RNA polymerase simply moves along the DNA and transcribe the structural genes. Okay, so once we add lactose to the medium, then what happens? That lactose enters into the cell, and so because there will be some number of very less quantity of permease molecule and very less quantity of beta galactosidase. So these two proteins always exist in the cell whether the lactose is present or not. So if the lactose is present, then the concentration of these proteins will be high. If the lactose is absent, then the concentration of these proteins will be very low. So that is the difference. So here the difference is it is not completely switching off the genes or not completely switching on the genes. Okay, so it is just regulating the genes, the lactose itself regulating the gene expression. Okay, so that means the genes are almost in switched off form, but it increasing the expression rate. The rate of the expression is increased by the presence of lactose. So if there is no lactose, then the rate of the synthesis of or expression of these structural genes will gradually come down to almost zero state, not completely zero state. Okay, so that we have to remember one more important point when we go into the maximum details of the lactose operon structure. Okay, the lactose operon functioning. Okay, so now because of that lactose that enters into the cell, and because of less quantity of beta galactosidase enzyme, so that beta galactosidase convert the lactose into allolactose. So allolactose is in fact that is so here the hydrolysis product, it is not the hydrolysis product of the lactose. In fact, the glycosidic bond is shifted from 1,4 position to 1,6 position, that's all. Okay, this allolactose acts as a potent inducer. Okay, the lactose acts as an inducer, and as well as the allolactose also acts as inducer, but allolactose is more potent inducer than the lactose. Okay, so therefore once the lactose enters the cell, it converted into allolactose, and because of that allolactose, what happens? That a repressor molecule binds the allolactose. Okay, So that this binding brings some conformational changes in repressor structure, so that the repressor structure gets changed, and that changed repressor structure is unable to bind the operator sequence. Okay, so that means the complex of repressor, it's with its inducer molecule is an inactive complex. So repressor becomes inactive by the binding of inducer molecule. So inducer repressor complex is inactive. So because it is inactive, it cannot bind the operator site. So in earlier case, the repressor is already bound to the operator site in the absence of lactose. So now what happens, that lactose or allolactose comes and bind to the repressor which is already occupied the operator site. So once it binds to the repressor, the conformation changes in the structure of repressor. Now that repressor dissociate from the operator site. So once the operator site is cleared from the repressor molecule, then what happens, the RNA polymerase now it is free to bind the promoter site. It binds the promoter site and it start transcription of structural genes. Okay, once the polycistronic mRNA is synthesized in this transcription process, that undergoes a translation process in which we will get the organism get three proteins, corresponding proteins of the structural genes, and they will participate in the metabolism of lactose. Okay, uptake and metabolism of lactose. So this is what the lactose operon. And so now in today's class, and we have already also discussed about the repression process, and I will not go into the details of repression process once again, because we have another topic in the next video lesson that I am going to cover, and that is the attenuation process. To understand that attenuation process, I will explain the repression process briefly once again, and then we will go into the details of attenuation process. Okay, so because the tryptophan operon is regulated by two mechanisms. One mechanism we have already discussed, already studied in the previous video lesson, and the second expression regulation is transcription attenuation process. I am going to cover that syllabus, that topic in the next video lesson. So let us skip this repression process and enter into the next process, which is catabolite repression. So the next topic is catabolite repression. So first of all, we try to understand the name of this topic, that is catabolite repression. So this is also a repression process in which something will be repressed or inhibited or stopped or switched off. So repression means simply stopping. Okay, something is being repressed by what? A catabolite. Okay, this is what the catabolite repression. Catabolite repression means, catabolite means anything that undergoes catabolism. Catabolism means nothing but breaking down. Okay, in the metabolism we have two processes. Metabolism is defined as catabolism plus anabolism. So here, so metabolism means catabolism plus anabolism. Here catabolism means breaking down and anabolism means synthesis, biosynthesis. So you have already studied these catabolic and anabolic process during the metabolism. Okay, in metabolism paper you have all the catabolisms and anabolisms. So if we take example of carbohydrate metabolism, you will study the catabolic process and anabolic process of carbohydrates, like glycolysis, TCA cycle, and glycogenolysis. All this comes under catabolism, okay, in which the glucose and other sugars are hydrolyzed or broken down, okay. That is catabolism. And anabolism means synthesis. From simple molecules, complex molecules will be synthesized. They are just simply the biosynthetic pathways, like gluconeogenesis. From non-carbohydrate precursors, we will get glucose in a process known as gluconeogenesis. From glucose, from excess glucose, uh, what happens, the glycogen will be synthesized to store the glucose, okay. That is also an anabolic process, okay. Like that, we have several different metabolic pathways. Some comes under catabolism, which involves breakdown, and some comes under anabolism, which involves biosynthesis, okay. So here catabolite mean that anything that undergoes breakdown, okay. If the organism take some material from the outside as a food material, then it becomes a catabolite. So the sugars we are going to add to the growth medium of E. coli cell, uh, they are known as catabolites, okay. So lactose, suppose if we take that this already well discussed example, that is lactose. Lactose is a catabolite, and if we add glucose, then glucose is also a catabolite, okay. So there are different types of energy sources for the bacterial cells. You can add glucose, you can add lactose, you can add mannose, you can add sucrose, or whatever it may be. So then if the organism is capable of utilizing those catabolite for the production of energy and material, then you can use it, okay. So different kinds of catabolites or sugars can be used as a source of energy, okay, for the E. coli cells, okay. So that is what the catabolite. Here, that the main observation that we have to do is, if we add glucose and lactose, both the sugars, to the medium at a time, so same time simultaneously at a particular time, we are adding both the energy sources like glucose and lactose. So then what happens, which uh, which carbohydrate will be preferred as the energy source by the E. coli cell? So that is the point that we have to discuss in this catabolite repression process. So in fact, if the glucose is present along with any other sugars which are known as secondary sugars, okay, then what happens, the bacteria always prefer to use glucose as the primary energy source, of energy. And once the glucose is depleted, then only it shifts its attention towards the remaining secondary sugars. So the first priority is for glucose, and the second priority goes to the remaining sugars except the glucose. So that is what here the catabolite repression. So that means the presence of glucose is repressing the utilization of other secondary sugars, even the secondary sugars are present in the medium along with the glucose, then in that case also still the organism is unable to utilize those secondary sugars, okay, although they are present in the medium, because it depends on the primary sugar. Okay, so if the primary sugar is there, that is glucose, then it doesn't matter whether the lactose is present or absent, simply it utilizes glucose. If the glucose is depleted, then only it will see whether the lactose is there or not. If the lactose is there, then it utilizes the lactose. If the lactose is absent, then it won't utilize, it won't switch on the lactose operon. So that simple thing that we are going to see, discuss in this video lesson. So the mechanism of regulation of lactose operon, which is under the influence of glucose. So that we are going to study in this video lesson. That is nothing but catabolite repression. Okay, so the E. coli cells always prefer utilization of glucose as primary sugars, okay, over lactose, arabinose and other sugars. They are called secondary sugars. And this kind of regulation of lactose operon by glucose is known as catabolite repression. So catabolite is here that glucose is repressing the lactose operon. So therefore the organism utilize glucose only. Okay, so that is what the catabolite repression. Once the glucose levels depletes, so that means if it utilizes the glucose, then what happens, gradually the glucose levels drops, and once the glucose is completely depleted, then the operon of other sugars will be activated in the presence of their respective sugars. Okay, so if we add glucose plus lactose to the medium, then first of all it utilizes the glucose and repress the lactose operon. Okay, once the glucose concentration goes down and complete glucose is depleted, then it will look at the lactose, and then it start utilizing the lactose by switching on the lactose operon. Okay, so that is the basic concept of catabolite repression. And so here the very simple doubt that may come across in our mind when we discuss about the catabolite repression. Okay, so that is how the organism is going to sense the presence of glucose or absence of glucose. Okay, so whether the glucose is present or not, how it is going to sense, and then how that is going to repress or activate the lactose operon. Okay, so here we have an sensing molecule which acts as an indicator of the glucose presence and absence of the glucose. Okay, so that indicator is the cyclic AMP. So the effect of glucose is mediated by presence of cyclic AMP. So cyclic AMP is a co-activator here, and cyclic AMP, as we all know that we know what is AMP, adenosine monophosphate. It is a nucleotide that comes from ATP. So we all everybody know what is ATP, and from the ATP, if we remove two phosphates, okay, so this is ATP, and if we remove these two terminal phosphates like in the form of pyrophosphate, then what happens, we will get this one, ATP. So that is adenosine monophosphate. Okay, clear? So now there is a specific enzyme known as adenylyl cyclase. This is not an ATPase, this is adenylyl cyclase. So this adenylyl cyclase synthesize cyclic AMP. So this is normal AMP, but it is cyclized. The phosphate is being cyclized. So that cyclic AMP is synthesized from ATP. Cyclic AMP in many cases you may have already came across with the functioning of cyclic AMP. Cyclic AMP acts as a secondary messenger in signal transduction. When you study the signal transduction process in cell biology paper, you will know the importance of cyclic AMP. Cyclic AMP acts as a messenger molecule, intracellular cellular messenger molecule. Okay. So here from the ATP, the cyclic AMP is synthesized by adenylyl cyclase. So cyclic AMP structure is this one, and in this structure we have 3' and 5' OH groups. So the phosphate, we have 5' phosphate, and that 5' phosphate will interact with the 3' OH group. So that is why it is known as 3', 5' cyclic AMP. Okay, so it is cyclized between 3' hydroxy and 5' hydroxy. So the phosphate is being cyclized. So this is the cyclic AMP. So one important thing we have to remember is, in the presence of glucose, the cyclic AMP levels rapidly decreases. So if a small quantity of glucose is there, so then the cyclic AMP levels rapidly decreases. Okay, and if the glucose is absent, then cyclic AMP levels goes up very rapidly. Okay, so that in that way the organism or the cell is going to sense the presence and absence of the glucose. If the glucose is there, then cyclic AMP levels will be low. If the glucose is absent, then cyclic AMP levels will be high. So this is what the mediation process by the cyclic AMP. Okay, so now when we look at the complete structure of lactose operon, so in fact when we discussed about the lactose operon, we have shown only these structural genes and operator site and promoter site and lac gene. We did not mention about this cap binding site. Okay, cap binding site, this we did not mention because I don't want to confuse you at the time of discussing about lactose operon. So if we talk about this particular site during the lactose operon itself, so that will confuse that will complicate your understanding. So that we have removed, we have not discussed about this thing. So but now it is essential to understand the catabolite repression process. So we have one particular site, that is a cap site. Okay, catabolite activator protein. Okay. So now what happens here, so this cyclic AMP, the cyclic AMP, in fact it is bound by a protein, bound by a receptor protein, that is cyclic AMP receptor protein, which is also known as CRP. Okay, in the absence of glucose we will have cyclic AMP levels, and this cyclic AMP will bind CRP. Okay, so this CRP with the cyclic AMP binds to the cap site, of the lac operon. So lac operon is having one more additional region, that is cap site. Okay, so this cap site is bound by cyclic AMP and CRP protein. Okay, so one more thing you should remember is, this cap site is located downstream to the promoter sequence. So here the operator is downstream to the promoter. That is why it is obstructing the movement of RNA polymerase, but the cap site is upstream to the promoter site. So that is why it won't obstruct the movement of RNA polymerase. In fact, it will increase the affinity of RNA polymerase to the promoter site, with the promoter site. So that is why it is acting as activator and repressor acting as repressor. So it will switch off the genes when the repressor binds to the operator, then the genes will be switched off, but the cap site is bound by CRP protein, then it activate the promoter. So two different functions. Okay, one that repressor is stopping, and cyclic AMP along with CRP is that is encouraging, activating the genes. Okay, so now when we study 4 different cases, okay, you have glucose, suppose medium, and lactose medium. So 4 different cases. In first case only lactose is there, in second case only glucose is there, in third case nothing is there, and in the fourth case both the glucose and lactose are there. So what happens in all these 4 different cases, we will look at and discuss each and every in detail, so that you can easily understand. Only thing you have to remember is, here the glucose is not there. In first case what happens, let us look at minus glucose. So that means glucose is not there. If the glucose is not there, then what happens, cyclic AMP levels goes up. So if the cyclic AMP levels goes up, then what happens, CRP plus cyclic AMP complex, it is an activator protein. So CRP and cyclic AMP levels increases. So that cyclic AMP level, the cyclic AMP along with the CRP, that binds to the cap site. So this small one is the CRP protein, and this one is the cyclic AMP. So cyclic AMP binds CRP, and that associated, that complex binds to the cap site. Now it acts as a activator. Okay, so now we have activator. Okay, so and the second point is lactose is there. So because the lactose is there, then what happens, that lactose binds to the repressor and make that repressor inactive. Okay, so now that repressor cannot bind to the operator site. So we have a situation like operator is free, so no inhibition, and cap site is active, bound. Cap site is bound with CRP, cyclic AMP complex. So now activator is there and inactivator or repressor is absent. So therefore what happens, the transcription of lactose operon takes place. So that means in the presence of only lactose operon will be active, and that utilizes that lactose only. Okay, so and in the second case only glucose is there but not the lactose. So what happens, glucose is there, therefore the cyclic AMP levels decreases, and no CRP binds the cyclic AMP. So then the cap site is unoccupied, so there is no bound protein or activator at the cap site. And second point is lactose is also absent, so that means that operator site is occupied by the repressor. So then this positive regulator is absent and negative regulator is present. So therefore there is no expression of structural genes. So lactose operon is completely shut down. Okay, so that is the second case. And in the third case, both the glucose and lactose both are absent. So then what should happen, the lactose operon must be repressed because there is no lactose. Okay, so here because the glucose is absent, the cap site is occupied, activator is there, but because the lactose is absent, the repressor is also there. So we have repressor and as well as the activator, both are there. So therefore what happens, because of the repressor, even though activator is there, because of the repressor that is obstructing the movement of RNA polymerase, so then the lac genes are not at all expressed. and the fourth case both are there. So this is the actual catabolite repression. This case is considered as actual catabolite repression process. Okay, both glucose is there, lactose is there. Okay, so because the glucose is there, then what happens? Cyclic AMP levels will be less, so therefore the cap site is free. Okay, no CRP binds to the cap site. But because the lactose is there, then what happens? The operator site is also free, because that lactose binds to repressor and make that inactive. Okay, so now what happens? Here the activator is not there, and at the same time the repressor is also not there, nothing is there. So then what happens? The RNA polymerase is unable to bind to promoter, because the activator is not there, but the operator is also unoccupied, so a very low level of expression takes place. Okay, very low level of expression takes place. So if the cap site is occupied, so then high level of expression takes place. Okay, so that is what the role of the cap sites. Cap site is there, that enhances the regulation, and if cap site is unoccupied, no activator is there, so then the expression is decreased. Okay, so very low or basal gene expression takes place. So very small quantity of beta galactosidase, permease and transacetylase synthesis takes place. So because of that less concentration only, whenever we add the lactose to the medium, that allows a small quantity of lactose into the cell, and that lactose will be converted to into allolactose by the less concentration of beta galactosidase. Okay, so this is what the original process that takes place. And so catabolite, here the glucose acts as a primary sugar, and that repress the operons of secondary sugars like lactose. So in the presence of glucose and lactose, then what happens? It prefers glucose only, and as long as the glucose is there, it continuously repress the lactose genes, and once the glucose levels goes down and glucose is completely depleted, then only it switch over to the lactose operon, and it start utilizing lactose by expressing structural genes of lactose operon. So this is what the catabolite repression process. Okay, hope you understand well, and thank you all.